Hang in there, one more. So thank you, Amy. Thanks for inviting me to the organizers. And I am Steve Averbuch. I work at Bristol Myers-Squibb, but today I'm actually representing not Bristol Myers-Squibb, but a much broader consortium of pharmaceutical and diagnostic companies that came together well over a year ago to try and solve a problem that you've heard a lot about today. I guess we can call it the PD-L1 problem. So under the theme of expanding opportunities for collaboration, I'm hoping in the next few minutes to share with you the story of the Blueprint team. That is an effort of this consortium to address a problem that was beginning to give a lot of people pause and real concern about chaos that was going to take place in the marketplace. So as we heard around from our FDA colleagues and others, when you're developing a drug that's dependent on a biomarker, there are regulations around what's required to establish a companion diagnostic. The current state of in vitro diagnostic, therapeutic diagnostic co-development is dictated by regulations, and each of the companies that are developing drugs for defined populations or populations defined by biomarkers in large part need to heed the regulations and the guidelines to establish the parameters by which the patients are defined by those biomarkers. So the landscape is one where independent and unique test development takes place in the context of each therapeutic product. Multiple drug and diagnostic complementary or companion diagnostic pairs can enter the market in parallel or in sequence. And this is not unique to PD-L1, but certainly has already taken place for biomarkers such as EGFR, BRAF, and others. So there's a complex challenge for testing and decision-making in the clinic when you have multiple opportunities for different diagnostics and different therapeutics. And at the end of the day, with the patient in mind, there's potential harm if inappropriate tests are used to make treatment decisions for which they were not intended. So we've already had multiple presentations today explaining the role of the PD-1 pathway as a checkpoint marker for T-cell exhaustion. And I only bring up this slide to remind you that the PD-L1 ligand PD-1 receptor interaction is incumbent to the mechanism of action for the PD-1, PD-L1 antibodies that were designed to interrupt that ligand receptor interaction that caused immune suppression through T-cell exhaustion. So therefore, it made teleological sense and was very plausible that PD-L1 had to be present either in the tumor cells or the immune effector cells for these drugs to work. There was also a literature historically showing that PD-L1 in many tumors, but not all, had prognostic and generally negative prognostic significance. So that was the starting point. It was an extremely plausible hypothesis that the PD-L1 biomarker was going to determine patients for whom the PD-1 and PD-L1 directed drugs, antibodies, were going to work. As a result, multiple sponsors, including BMS, Merck, AstraZeneca, and Roche Genentech initially, began to develop their antibodies specific for the PD-1, PD-L1 axis. In conjunction, they partnered with diagnostic companies to develop PD-L1 immunohistochemical assays as a one assay, one drug paradigm. These assay scores and interpretation guidelines were developed to identify responding populations for the unique drugs and the unique biological hypotheses. So it's important to recognize that each of the companies involved here had very different strategies and underlying hypotheses that were driving their phase 2, phase 3 registrational programs. The companies had differences with respect to the utilization and definition of the biomarker as to whether it was going to be a companion, i.e., as per regulatory, as FDA definitions, essential for the effective and safe use of the drug, or whether it was going to be complementary, which is not an official FDA designation, but it has been accepted as a definition that whereby the information is useful in the management of patients. There were issues around confidentiality, IP constraints, contractual obligations that required that assays be developed within firewalls, even within a single diagnostic organization. So as multiple pharmas partnered with a single diagnostic organization for different assays, within that diagnostic company, there needed to be separate firewalled activities for the development, registration, manufacture, and commercialization of that respective diagnostic. So this was the landscape in which we were facing. And as you heard from Myers-Squibb earlier and others, at least 4 assays have been analytically validated and used in clinical studies to test the PD-L1 hypothesis related to its status and its association with therapeutic outcomes with the utilization of PD-1 or PD-L1 directed therapy. The 4 companies I identified are here with their respective anti-PD-1 or PD-L1 antibodies. 2 diagnostic companies were involved, DACO and Ventana. 4 different antibodies, 4 different scoring methods. So you had 4 different drugs, 4 different antibodies, all against the same target. And as you heard earlier, there was a lot of concern about the confusion that this was going to cause in the community of practice as these drugs became available. And I'm not shy about sharing many comments from FDA colleagues through conversations and from the podium that their concern was this was a train wreck coming. And one individual in particular kept telling me PD-L1 is giving her a headache. So this was the problem. We've talked a lot today about the biomarker, and particularly about the PD-L1 biomarker. And I have to just take a moment and digress and remind everyone that biomarkers are not diagnostics. And we talk cavalierly about biomarkers, but it's absolutely critical that we impose the same, not the same, but the highest standards of quality and performance to the biomarker as we do to the therapeutics. So therefore, we need to ensure that the diagnostic, which is not just an antibody, but is a whole system, and in this case, for immunohistochemistry, it involves an antibody. I already told you there are 4 different antibodies. Reagents, each of the assays has a different reagent system, a different platform, the machine that you put it into, the software that controls that machine, and the human factor, the pathologist who's reading the slide and interpreting the score of a very heterogeneous, very variable tumor sample underneath their eyes through the microscope. That whole system is what needs to be evaluated and determined with respect to its reproducibility, reliability, and accuracy. So this was our problem that we faced as a community. Each biopharma company's agent and assay development was different and induced complexity for patient management. The hypotheses and patient populations under study for market approval were all different. Each biopharma company had partnered with a diagnostic company to independently develop an analytically validated IHC system measuring PD-L1 expression. And the relationships between the efficacy endpoints, the patient populations, the defined inclusion criteria were different across sponsors and assays in randomized studies and in non-randomized studies with respect to the type and timing of the biopsy, the cell types of interest, the scoring method, and the cutoff. So as I mentioned, 4 different assays, each in their own swim lane, was creating a potential train wreck. And a perception in the community that these assays were highly discordant. A number of publications have come out, especially in the last year, trying to compare the different antibodies and assays, although the health warning here is that these are being generated from academic laboratories, usually using laboratory-developed tests and not necessarily employing the standardized, validated biomarkers that have been developed by Academies and by Ventana. Keith Kerr from Aberdeen, in his paper in the Journal of Thoracic Oncology, among many others, proposed that a multi-center international standardization effort was required to address all of the concerns I've alluded to around the testing complexity upon approval of one or more drug or diagnostic. Well, I'm very proud to say, as part of this industry pre-competitive collaboration, we attacked this problem well before the marketed approval of any of the drugs or diagnostics. Recognizing that there was a different test for each drug is impractical, and that using one test for every drug was going to be unpractical, and recognizing that there was potential harm to patients if the FDA-approved IVDs and drugs were cross-matched by end users in the absence of cross-validation, interchangeable information around the different assays. The pharmas involved and the diagnostic companies involved came together on their own volition to address this problem. This happened in advance of a workshop in March of 2015. Several of us began to have discussions among the leadership of the pharmas and then subsequently the diagnostic companies. We had a series of teleconferences, and I remember the first teleconference in which we came together to discuss the potential for a pre-competitive collaboration. And the first thing that was said by the members participating in that teleconference is, this is for the patient. We need to do something to establish standards and understanding around the diagnostic assays that will enable the community of medical practices, especially the pathology community, the oncology community, the laboratory services community, to figure out this level of complexity. The problems were highlighted at this FDA workshop in March, using PD-L1 as a case study for the complexity of multiple assays linked to multiple therapeutics, building public awareness of the issue, to foster a public examination of the problem, and to offer potential solutions. So the Blueprint team came together and presented at this FDA workshop with a proposal, hence called the Blueprint. And this was a collaboration among the 4 pharmas seen here, the 2 diagnostic companies seen on the slide, Dako and Ventana. It was facilitated at the forum by AACR and facilitated throughout the remainder of the year through AACR. And I have to acknowledge Rasika Kalamagan, who's in the audience, for her efforts in being the driver and the facilitator for this when she did work for the AACR. And the FDA and EMA were independent, neutral observers to the process. We engaged an independent, honest broker, and that was the International Association for the Study of Lung Cancer, their pathology community, to collaborate with this consortium to evaluate the assays in a cross-comparative study. The proposal was the goal was to agree and deliver via this cross-industry collaboration a package of information and data upon which the analytical comparison of the various diagnostic assays may be conducted, potentially paving the way for post-market standardization and our practice guideline development as appropriate. One of the undercurrents here was taking the HER2 situation as a poster child. It took 12 years to get standardization of the HER2 assay into ASCO-CAP guidelines. One of the hidden goals of this, or the sub-theme goal, if you will, was to short-circuit that process by 1 or 2 years rather than 12 years to get us to a point of standardization. The intent was to only look at the analytical performance. This is not a cross-trial comparison. It's not to use clinical data, and it's not to violate the boundaries of IP and the proprietary information that is still part of how each pharma, each diagnostic concern needs to develop their product for their business needs. So it was only restricted to those tests that were on the PMA pathway to approval, not to compare with LDTs. These were the tests that are being used currently in the clinical trials for registration. We insisted that there would be no delay to pivotal studies and patient access to critical new therapies, and we wanted to focus on non-small cell lung cancer as the first case study. So the ultimate objective was to deliver this package of information to the medical community. Each of the members in the Blueprint project had defined roles and responsibilities, which are listed here. The initial aim to characterize these 4 assays, the details are shown here, with an initial focus on non-small cell lung cancer. Pathologists from external organizations were engaged to provide independent interpretation of slides that were stained by the respective diagnostic companies and then exchanged across the diagnostic companies for evaluation by the respective pathologists. And then the ISLAC pathologists came in and did the score verification. This was designed as a 2-phase study. The first phase has been completed. This shows the details of how the staining was done by each of the respective diagnostic companies, exchanging the information, collaboration between the pathologists and the biostatisticians to have statistical robustness. And this face-to-face meeting was held in early November to review the data and to have a slide review. From that evaluation and then rereading of the data and additional statistical validation, we plan to present these data at AACR in April in New Orleans. So in summary, I hope I've left you with a case study that is a good example of how we can move the field forward when we have some of the complex problems that are facing us as a community for the advent of newer immunotherapies. And perhaps this can apply beyond just immunotherapy for future policy approaches to cancer. Blueprint is a successful pre-competitive collaboration to evaluate these assays for PD-L1 diagnostics that are important for patient management. We are considering to embark on the phase 2 for additional rigor, but I think we will await the publication and the presentation, the publication at AACR to determine whether or not additional studies are needed or whether we've accomplished our goal through the phase 1 portion of the project. And we will be fully transparent with all the findings from all the companies at our AACR session in April. So with that, I want to identify all of the people that participated. It's been an extreme privilege for me to be a part of this and to see us move the field forward for the benefit of patients and the medical community. Thank you.